Okay, so I'm from Jean Picou's lab, and I'm going to represent the lab because Jean's out of town right now. One half of the lab has traditionally been more involved in synthetic biology and the development of tools for synthetic biology, but a lot of that work has now moved over to a startup company that Jean has started. So what I'm going to talk about is our work on the study of cell cycle regulation, and we use the budding yeast as a model system to study this. Our approach is to take a modeling experiment cycle approach to study this. And in terms of the modeling, we do a lot of that in collaboration with John Tyson's lab over in the biology department. And the approach is to use models to try to understand the molecular dynamics underlying cell cycle transitions and develop these very complicated models to help explain what's known about the molecular dynamics, and then use the experiments to fill in gaps about the molecular dynamics using molecular and synthetic biology approaches to get quantitative data that we can then put into the model and parameterize it. And then what we do is test predictions of the models by perturbing these systems, mostly through mutations in the budding yeast, and then looking at the phenotypic outputs of those perturbations and seeing if the model correctly predicts what the outcomes are. And then we feed back into the models to better refine the models and represent the new data that we generate. So the overarching goal of this is basically to understand the network dynamics that lead to irreversible transitions between the different stages of the cell cycle, and also some of the quality control mechanisms, otherwise known as cell cycle checkpoints, that inhibit these transitions when there are problems in the previous phase of the cell cycle. By taking this approach, we hope to better understand the dynamics of cell cycle regulation, and a lot of this will feed into our understanding of cell cycle changes that occurred during embryonic development, tissue homeostasis, and when the cell cycle is perturbed in diseases like cancer. That's it.